Welcome back. In today's class, we will be learning about sodium potassium pump, which is the best example for active transport of metabolites into the cell. This happens under the influence of electrogenic transmembrane ATPase enzyme. This happens under the influence of ATP, where ATPase enzyme is involved to hydrolyze them and facilitate the diffusion. This ATPase enzyme facilitated mechanism was discovered by Jens Christian in the year 1957. Sodium potassium pump enables us to understand how sodium exit the cells and how potassium ions enter into the cell is with an equilibrium maintained between them is all about your sodium potassium pump. As I told you, it is the best example for ATP facilitated diffusion. It is an active transport mechanism which involves under the presence of ATP molecules. This sodium potassium ATPase enzyme facilitates the diffusion of nutrients into the cell. As there is enormous amount of sodium into the cell that has to be released outside, and then potassium should be brought inside the cell. It is always in such a way that 3 sodium molecules will leave the cell and 2 potassium molecules will enter the cell. That is why we have written it as 3 sodium to the cell's exterior, as in 3 sodium molecules exit the cells, 2 potassium ions enters the cell. This happens under a super standard equilibrium maintained by the cell. This is a p-type ion pump because the ATP interacts with the transport protein, causing it to phosphorylate and change its conformation. In the diagram, I would explain how the conformational change of the intrinsic proteins that are present in the cell membrane will be explained. This is a protein-based enzyme which is present in the membrane of the mammals. This electrogenic transmembrane ATPase facilitates sodium potassium diffusion, sodium exit and potassium entry into the cell. As we look into the sodium potassium ATPase structure, it is having several subunits inside it, alpha subunit, beta subunit, and several proteins. And it has a function of maintaining a sodium and potassium ions into it. So maintaining the equilibrium of sodium and potassium ions, maintaining its resting potential, importing nutrients into the cell, exiting the unwanted metabolites from the cell, regulating its cell volume, all that has been maintained by ATPase enzyme. Then comes its regulatory actions. It maintains the activity of the cell, enzyme trafficking, gene expression, and phosphorylation is facilitated by sodium potassium ATPase enzyme. As we see, if this is the structure of your cell, there will be 3 molecules of Na, that is sodium, leaving the cell, 2 molecules of potassium entering into the cell under the influence of ATPase enzyme. ATPase will hydrolyze ATP to ADP and inorganic phosphate. This is a brief explanation of how sodium potassium pump works. As we know, the cell membrane is made up of lipid bilayer. This is the hydrophilic head and hydrophobic tails. In between them, they have something called as embedded proteins. This exchange the nutrients in and across the cell. 3 molecules of Na will leave the cell. 2 molecules of potassium will enter into the cell. This is the inside of the cell. This is the outside of the cell. This is the cytoplasmic content. This is the inside, interior of the cell, where molecules left is sodium and the molecules entering is potassium. And this happens under the currency of your ATP, where ATP is hydrolyzed into ADP and inorganic phosphate under the influence of ATPase enzyme. To be more particular, it is sodium potassium ATPase enzyme. The function of these sodium potassium pump stabilizes the ion motility in and out of the cell. Having too many cells inside the cell would affect its metabolic activity. So regulation of exit of the molecules is done, and then entry of the potassium ions is regulated. Many secondary active transporters are present, like your protein, which exports sodium potassium outside the cell and also regulate the uptake of the mineral nutrients into the cell. Sodium potassium pump maintains the cellular osmolarity. This osmosis of the cell can be maintained regulating sodium potassium pump. This maintains, adds on to the active metabolism of the cell. This extracellular signaling is carried out by the intervention of your sodium potassium ATPase enzyme. It also controls the cell state, equilibrium of the charges present, and then there are many drugs that are designed as in sample with sodium potassium. There the drug that is designed will mimic sodium ions, will mimic potassium ions so that it can enter the cell, and detoxifying metabolites like it, they will mimic sodium ions so they can exit the cell. This is how exactly a sodium potassium pump looks like. If you have a question like why only 3 molecules should leave the cell, 2 molecules should enter the cell, the conformation of the intrinsic protein present is in such a way that it has 3 conformation for the sodium molecules to fit in. So from the cell interior, sodium molecules will go and adhere there, and then ATP molecule is present. This ATP is nothing but adenosine with 3-phosphate. That is what is adenosine triphosphate. This will attach to the sodium and then they will move across. Under the influence of your ATPase enzyme, ATP will be converted to ADP, that is adenosine with diphosphate. And then this inorganic phosphate that is formed will attach to the ATP binding site, causes a conformational change in the membrane. So then these conformation changes like this, from this to this, so that the sodium ions will be liberated out of the cell. As the sodium ions are liberated out of the cell, it also takes in 2 potassium ions. So the vacancy for the ions are only 2. So 2 potassium ions will be brought in, and then this will be causing another conformational change to leave the potassium ions inside the cell. Please understand, these are the interior of the cell, these are the exterior of the cell. First, sodium ions binds to the proteins, which facilitates the diffusion under the influence of ATP. As ATPase enzyme will hydrolyze this ATP into ADP and inorganic phosphate, this inorganic phosphate will attach to the protein binding site or ATP binding site and changing a conformation. As it leaves away the sodium ions, it also takes in potassium ions. And then there is a conformational change that is happening again, where Pi and your potassium ions will be left out. So again, there will be new 3 molecules of sodium attaching here, another ATP attaching, so that the regulation is carried on further. So there are many strategies that these the sodium potassium pump follows in order to regulate them. So the first one is the resting potential. The resting potential is nothing but maintaining the potential to exchange the molecules. When there is low sodium into the cell, they switch to the resting stage where no active transport of sodium ions would happen. Next comes the reversal potential, when despite of your sodium and potassium having the same charge, liquid equilibrium must be maintained in the external or the internal concentration. Sodium potassium pump transports ion in and out of the cell, maintaining their function, maintaining their concentration. Thereby equilibrium in the cell can be maintained. Transport of the metabolites into or exit of the cell is being regulated. A cell volume is maintained by regulating the cell osmosis. Proper concentration of different nutrients will be assessed, and then the motility will be carried out. Signal transduction is excellently performed by your sodium potassium ATPase enzyme. Then this neuron activity state also stimulates the activities of neurons by adding on to the consistency of your sodium potassium pump active stage. So this is how a drug is administration, or an applicatory part of your sodium potassium pump is that they can be used in drug administration, where your drug will mimic potassium ions and your cell will take potassium ions into the cell, thereby taking the drug that is designed according to your potassium ions. So the drug can be administered into the cell. So this is all we have for sodium potassium pump. thereby how it regulates the cell, how it exchanges nutrients, how it uptakes the metabolites, what are the enzymes involved. This is all about your sodium potassium pump. Thank you.